Good afternoon and welcome to the fourth How TrueAllele Works. Today we'll be talking about genotype database and DNA investigation. So as you recall, DNA evidence on its own is quite straightforward. Suppose you have a single source evidence item from a crime scene shown on the left, and there's a single source reference who's a possible suspect, and their genotype values, say the allele pairs at all loci, match each other. If that's the case, then the likelihood ratio, or the match statistic, is just the rarity, or 1 over the probability of a coincidence, of seeing this genotype in the population. And this idea of comparing single source genotypes 25 years ago was taken by Peter Gill in the British government and turned into the idea of a DNA database. The idea here, starting from the bottom left of the picture, is that a single source sample from a crime scene has a very clear genotype from the data. It can be uploaded to a set of genotypes of thousands or hundreds of thousands, millions of different genotypes that all represent evidence from a crime scene. And on the bottom right, there are possible suspects who could be convicted offenders or ultimately in England became all arrestees, and their cheek swab pristine single source profiles are then uploaded as genotypes to a genotype database that can contain hundreds of thousands of individuals. And then, even though evidence may have been left at a crime scene and the crime not solved, and an individual who committed the crime is not a suspect by matching, as is shown in the center, between the evidence genotype database and the reference genotype database, cold cases could be solved by connecting crime scenes to suspects through the genotypes on their database. However, DNA mixtures, which have become prevalent and in many labs the main form of DNA evidence today, sort of stir things up a bit. Instead of having just one individual contributing their DNA to evidence, you could have 2, 3, 4, 5 or more people contributing their DNA, and that doubled at least the amount of interpretation effort and often far more than double and redoubled all the trouble involved in interpreting DNA. And the result was that the model of having single source DNA as the basis of a database didn't work as well. So one generalization that was tried was to say, well, a genotype has 1 or 2 alleles. Why don't we just use the same approach with mixtures, and let's look at the surface features from the data peaks. We can apply a threshold and look at the 1, 2, 3, 4, 5, whatever the alleles are that we think correspond to the peaks that we observe in the data, draw a line and say that list of alleles, that is now how we represent evidence. And so starting at the bottom left, what we see is that unseparated mixtures now become uploaded as lists of alleles to an allele database. So each item of evidence that's a mixture may have all the peaks that are represented at each locus. And the problem with that is that when comparisons are made to reference samples, there can be false positives, because it may be that by a simple investigative lead approach of doing a moderate stringency comparison that intersects alleles between different lists from evidence and reference may lead to more than one reference being found. In fact, it can lead to dozens or hundreds of references being found, because the match comparison is not that specific. It's not a statistic, it's just a heuristic for making a comparison between alleles. And as a result, in order to control the false positive matches, as we see on the bottom left, the uploads of unseparated mixtures from just lists of alleles had to be stopped or at least filtered at some level, to the point where today, with an American database that has over 10 million reference profiles, perhaps a 10% of mixture evidence makes it up to an unseparated allele database, and 90% of the evidence that's mixtures doesn't get there at all and can't be used to find possible suspects. This was quantified in a talk that I gave 3 years ago at the American Academy of Forensic Sciences in Atlanta on investigative DNA databases that preserve identification information. What we see in this figure is, let's look at the x-axis. Starting all the way on the right, we're looking at 13 CODIS loci, and that would represent that all 13 loci are hitting allele list evidence relative to a possible contributor. Moving to the left, 12 loci would mean that one locus isn't being used or isn't hitting, and so on as we move down to 11, 10, 9, and so on to the number of loci that are hit. The y-axis is giving the probability in two different ways. For the blue bars, it's showing the locus hit probability of how many loci get hit for the different cases of 1, 2, 3, 4, up to 13 loci. And more interestingly, the red line gives a false positive rate. If we read that red line starting all the way in the bottom right where 13 is, there's a false positive rate when all 13 loci are used. But when only 12 loci are used, that red line goes up of having 12 or 13. It's the sum of the blue bars at 12 and 13. At 11, the red line is showing the sum of spuriously hit profiles for 11, 12, and 13. As we keep moving down to the left, we see by the time we get to maybe 8 loci hitting, that tail probability with the point at the red line, which is adding up the probabilities for 8, 9, 10, 11, 12, and 13 of spurious hits, is giving a high false positive rate. Clearly, if you're lighting up 10% of a database that would have 10 million people on it, you don't want to get a million hits. So that's why rules are put in place, as shown on the left side of this picture, to control the upload and make sure that in an allele comparison database that most of these mixtures do not get uploaded to cause a high rate of spuriously hit reference samples. Fortunately, there are ways to approach this that overcome the problem that are based on not generalizing the database by use of having lists of alleles, but instead of having separated genotypes. So one concept that we've been talking about in these lectures is the idea that the data of a questioned item, shown here on the left as D sub Q, can be separated into the contributor genotypes. I've indicated them here by capital X1, X2, X3. These are the contributor genotypes that have combined to produce the data, and what we can infer from them are genotypes, perhaps with some uncertainty, and that's where probability enters. So on the right, I'm indicating that, for example, for the genotype for contributor 1, capital X sub 1, there's a probability, the probability of each value, each allele pair that X1 would assume. So each genotype can assume different values with probabilities that are conditioned on the evidence data, D sub Q. And for each, in this case, of 3 contributors and their genotypes, there are posterior probabilities, posterior because it's after the data has been seen, of what the genotype values would have as probabilities of being more or less likely. And so the idea is, with accurate computing and good statistical models, mixture data can be separated into contributor genotypes with associated probability distributions for each genotype at each locus. Now, a second idea is that after you've separated genotypes and you're back to a world of single source like DNA, single contributors, now the standard likelihood ratio from probability theory gives us the match information. We can combine the posterior probability of a genotype, shown in blue, from one evidence item, together with the probability distribution for variable y from another evidence item. It could be from a crime scene or reference sample, and that by multiplying them together to determine their match strength relative to the denominator, which is the rarity or the prior probability in the population and adding up all the different allele pair possibilities, genotypes and through their probability distributions can be combined in a likelihood ratio once you have them for single contributors. So this leads to the genotype paradigm that we've been talking about throughout this series. So on the top left in blue, we see that evidence from a mixture and its data can be separated into a number of genotypes known up to probability that are conditioned on the observed data. So mixtures are separated into genotypes and their probabilities. Now, a different evidence or reference item, in this case a evidence item, is shown in red for some subject or some sample data, D sub S, is similarly split into its genotypes and its probabilities. And that's the genotypes and the probabilities are shown in the center. And the second step is after data from mixtures, kinship, or whatever source of genetic information has been separated into genotypes, then they can be recombined into match statistics. That is, one genotype from one item, from Q, say the first contributor, and a second genotype, say the third one, from item S, are shown in the arrows as recombining to form a match statistic. And so that's the basic concept here, is to always reduce evidence down to single contributors with uncertainty represented as probability, and then look at pairs of them to combine them back into DNA match statistics. So that enables a genotype database. And now the problems that we saw before on the left half of the picture are gone. If the genotypes are separated, then they can be uploaded as single contributor genotypes. And those genotypes can now then be compared on a database up in a cloud somewhere with reference genotypes shown on the right, and there's no issue. And in fact, a likelihood ratio statistic is calculated that can give us a lot of information, and we'll need to rely on that to be accurate. But what you can see is that the yellow individual shown on the bottom left having contributed to crime scene evidence and shown on the bottom right as a suspect can be uploaded as genotypes on the top level and then matched compared with the statistic produced. Now, we need to rely on this statistic. From that same talk, we also showed likelihood ratio information from separated genotypes and how that produced accurate match results. So on these mixtures, on the right side, we're seeing the results of a sensitivity comparison with the true positives as the mixture items were compared with the true contributor genotypes that were known. And then on the left side in red, we're seeing the true negatives rejecting the matches or identifying mismatches and showing the specificity. The axis is in log likelihood ratios, and the average sensitivity in the study was 10 to the 15th, or about a quadrillion, and the average specificity was 10 to the negative 22nd, which is a very small number, like 1 over sextillion or so. And so there's a very good separation between true positives, true negatives, and that's relied on in a database to have accurate match statistics. As a result, there are all kinds of databases that can be developed. For example, here I've replaced the right side of the picture with an evidence database, possibly the same as the one before, and now we're comparing crime scene genotypes that have been separated, shown on the bottom left, with separated genotypes from a second crime scene. And when all of those genotypes are uploaded to the database, then evidence genotypes can be compared against evidence genotypes, and matches can be determined, as shown in the center, with that same likelihood ratio formula for general single contributor genotypes that we've seen throughout. And this is a way of having a database make automated case-to-case comparisons on 100% of the evidence without any restriction of what can be compared. Another use is to have a quality assurance database in the laboratory, where here the right side of the equation is now a personnel database, where single source reference samples from laboratory and police personnel are uploaded to a within lab genotype database. And now if there is some contamination or some other issue on the evidence side on the left, then that can be identified and dealt with, because it's just one more matching process of the same evidence, whether it's evidence to find case-to-case matches, evidence to reference to find likely suspects, or evidence to personnel to identify contamination problems. So I'd like to give an example of the use of databases in a case I was involved in this year in England, and this is a rapist from Southampton who committed a stranger rape on New Year's Eve around 3 in the morning. A woman was walking home and was sexually assaulted by a man she didn't know at this playground in Southampton in southern England. A rape kit was collected, and as the report read, there was a trace amount of semen detected on vaginal swabs submitted for DNA profiling. The DNA profile contained limited information, so a speculative search was done on the United Kingdom's National DNA Database, the NDNAD, and the search came up with 13 profiles, one of which was a person who was known to reside in the Hampshire area. However, due to the low profile and the incomplete nature of the data, the lab felt it was not suitable for routine statistical evaluation. So I'll quickly walk you through the genotype separation process, where proper quantitative modeling of all the variables and uncertainties and artifacts can use all the data. Here are quantitative peak heights at the locus D21. This data came from England, so the kit used was SGM+, which is a 10 locus kit. And here we see peaks for 28 and 32.2 that are taller. There's a lower peak below 50 RFU for a 29 allele for D21. And the sort of methods that were attempted in this case make less use of the data. They may be methods that apply thresholds, use dropout. They have fixed arbitrary parameters that don't engage the data and make the fullest use of it. So in a threshold method, over a threshold, peaks are labeled as allele events, so 28 and 32.2 are alleles that exceed a threshold such as 100 RFU indicated by red, but lower level peaks like the 29 that doesn't even reach 50 RFU would disappear, and in this case not much can be said when making a comparison. However, with a sophisticated computer model, then most every possible genotype solution can be attempted through statistical sampling, and how well each pattern accounts for the data can be determined by comparing hypothesized combinations of genotypes from different contributors, along with the variation that's determined from the data and stutter events, so on, to get a sense of what are better explanations. Here we see that one person's allele pair, which corresponds to the victim, of a 28 and a 32.2, indicated with a pair of blue rectangles that are relatively tall, explains some of the data, and another person's allele pair, who's not the victim, shown in orange, with a smaller amount, maybe a quarter of the amount of the major contributor, has an allele pair of 29 and 32.2, and when adding them together, we can see that the tops of the added together rectangles are reasonably close to the observed data. An actual computer would also account for stutter, relative amplification, and when making these comparisons, you actually use the variance as worked out from the data to determine how well the pattern actually does explain this data. And the result at this locus is a separated evidence genotype into a major contributor and a minor contributor. Here I'm showing the minor contributor. This is an objective genotype determined solely from the STR data without seeing a comparison reference for this contributor. And here we're looking at one contributor, one locus. And there are different possibilities. Out of the 100 or so possible allele pairs that we had before we started, this is reduced to maybe 8 possibilities at the D21 locus. None are really standing out, but this is a concentration of probability relative to what we had before with all the population allele pairs. So the computer has inferred this as an evidence genotype, and here is how it's represented as a posterior probability distribution. In order to form a match statistic, we need to compare that with another genotype. In this case, it'll be from a suspect, and then normalize with respect to how coincidental the genotype is by accounting for the population. So we try to answer how much more does the suspect match the evidence than a random person in order to determine the DNA match information at this locus. The blue bars that we're seeing are the same as before from the rape kit evidence. The brown bars are the probabilities of different Caucasian allele pairs at this locus. And you see that at some allele pairs, the blue bars are taller, at others, the brown bars are taller. When the blue bar is taller, that means that the evidence match is higher than a coincidental match, like at 29-29. And when the genotype probability from evidence is lower than the population, say next to it at 28-30, that's evidence against a match. In this case, at locus D21, all the way on the right, we fix on this allele pair 29-32.2 because that is the allele pair of a suspect. And so the math lets us largely focus at that allele pair and then make a comparison. What is the probability of the genotype in blue, 21%, divided by the probability of a random person in the population, here shown in brown is 4%. That can also be viewed as the probability of an evidence match divided by a coincidental match when you're comparing with a reference sample. And that ratio of 21-4 at the suspect's genotype, circled in red on the right, 29-32.2, is about a factor of 5. So relative to the minor contributor of this that's been separated out from the mixture, there's a match statistic of 5. And if we look at all 10 loci in SGM+, on the x-axis, here's the likelihood ratio, and the listed on the y-axis are the 10 loci. Some of the numbers are less than 1, for example, D3, D8, and FGA, indicating some support against an inclusion. But most of the numbers are further to the right, and when they're multiplied together, numbers like 7 times 2 times 4 times 5 and so on, accounting for all the data and not picking and choosing but multiplying all 10 numbers together, we arrive at an answer to the question, is the suspect in the evidence? And we can report, as we did, that a match between the vaginal swabs and Stuart Ashley Burton is 17,000 times more probable than coincidence. Now I'd like to turn to what happened with the database hits, because this has fairly general relevance for crime labs. So there were 13 hits, and I've shown them in sorted order. Left to right is the log likelihood ratio. I've indicated where 0 is in red and 4, or a statistic of 10,000, across the top. And they're sorted going from around -1 up to +4, or about 10,000. And what we see is that there seems to be a clustering where the other 12 match statistics from these database hits are kind of around 0 or 1 and just randomly varying. And then the question is Mr. Burton actually an outlier? Is he statistically different than these other individuals based on a comparison of the rape kit evidence, that minor contributor, to each of these 13 reference samples? So to address that question of how do we separate out these database hits, we can form a non-contributor distribution for the separated minor contributor from the rape kit evidence. And what this shows us is what is the distribution of log likelihood ratio of the match statistic information when compared with someone who did not contribute their DNA to this evidence, someone who is not actually this contributor. And one way to do that is by comparison with random genotypes. But what we see regardless is a bell-shaped light curve, and on the axis we have a log likelihood ratio values centered at around 3, -3. The actual mean is -2.7. So this is overall exclusionary. And there's a standard deviation of about 1.5. And you can see that right tail moving up to past 0 to 1 or 2. So it would not be unexpected to find individuals who produced a positive match statistic given the distribution of non-contributors. There may be people who did not contribute their DNA, and we see them in 12 of the database hits. So here again is the non-contributor distribution, and now what we're going to do is assess those 13 match statistics and their size in the context of a probability distribution that was determined solely from the evidence for non-contributors. In red, we see where the 12 hits were that were not Mr. Burton, and we also see where they lie along this bell curve of non-contributors. And if we go all the way to the right, out to a value of almost 2, just under 2 log units, just under 100, if we look at the tail probability to the right, which tells us what is the chance that an individual has that match statistic or greater, it never exceeds 1 in 1,000. So what that bell curve, that probability distribution of non-contributors is telling us is that it's not that unexpected to see these spurious hits. But when we get to Mr. Burton, shown in green, even though his match statistic is only a little more than 2 log units, it's a little more than 4 instead of less than 2, looking at the tail of that bell curve, that normal distribution, that tail distribution is 1 in a million. So the chance that somebody on this evidence would have a statistic that is at the level of 17,000, at the level of 4 log units or greater, is less than 1 in a million. So that extra information tells us that this would be a rare event, and this is a way to separate out database hits when you have more than one of them, like in this case, about a dozen. And the DNA and good police work led to a conviction. Mr. Burton pleaded guilty to the New Year's Day rape and was sentenced to 12 years in prison. He was also convicted at that time of having sex with a young girl, and he had had a history of violence in the past. There are other applications of these types of genotype databases, and I'll just go through a few. One is familial search. I won't draw the entire database architecture, just the sorts of genotypes that are being looked at once they're separated on the database. On the left, we can have any sort of evidence containing 1, 2, 3, or more separated evidence genotypes, and now the likelihood ratio for the match can make a comparison between single contributor genotypes and something else. And on the right, what I'm showing is inferred genotypes from relatives of reference genotypes. So for example, suppose there was a database of convicted offenders and the child shaded in labeled as C was on that database. One could build a genotype database of close relatives, father, sibling, child, so on, of that individual labeled C in blue. And those would be genotypes. There'd be genetic uncertainty, so they'd have probability, but one could build an entire database of the close relatives of of individuals who were suspects. And that way, if you did a comparison, automatically the computer database would be comparing separated evidence genotypes into their own single contributors relative to inferred family members, like a father or sibling, here shown as a father, and that would just happen automatically. There'd be no special need to run searches on partial matches. The genotypes of the relatives could be in a kinship database of convicted offenders and automatically search. Another approach is to compare missing persons with genotypes. The way this works is that we don't have the missing person, but we do have their family. And so we can infer, as we saw last time, a genotype inferred from reference relatives. Here that person's labeled X, and we show that there are some family members. The more family members, the more definite the probability distribution of their genotype. But that's just another genotype with some probability attached to it to represent the uncertainty. That's loaded up on the right side here in the picture of a database. Ordinary evidence, whether it's from mixtures or kinship or degraded DNA, whatever have you, is uploaded on the left side as evidence genotypes from single contributors, and then typical automated likelihood ratio comparisons can be made in order to find likelihood ratios between evidence and missing persons. This type of genotype database can also be used for mass disasters, where there are victim remains genotypes. They're typically single source, so one doesn't have to make a Solomonic decision about what do you do with mixtures from victim remains. And on the right side, comparisons can be made with kinship inferred genotypes from known family members shown on the top, or with genotypes from personal effects from a missing person. And again, there's no change in architecture, there's no change in math, there's no change in procedure. All that happens is that single contributor genotypes from whatever source, mixtures, charred remains, kinship, personal effects, all uploaded to a genotype database for single contributors, and then matching occurs automatically between them, computing sensitive and specific likelihood ratios. This idea of using a database as part of the work in DNA analysis can be taken to another level, as Dr. Kevin Miller's lab has done in Kern County. This is described in an article that we wrote called Kern County Resolves the DNA Mixture Crisis that appeared in Forensic Magazine last fall. The idea here is to take advantage of the matching power of the DNA database. The entire point of the architecture is that when the genotypes are separated from mixtures or families, the information is preserved. When they're uploaded to a database, the information isn't changed. When the matching between pairs of genotypes are made, it's using a likelihood ratio which preserves and quantifies the information. So you're essentially getting very close to what you'd be getting in ordinary casework, inferring genotypes and computing likelihood ratios, but it's all happening on the database. And that can tell you where the identification information is by focusing on the matches that are actually there and not on the vast majority of comparisons that don't produce a match. Most comparisons produce exclusionary information, which can be of interest, but usually not for the large-scale work the crime labs do. So in this model, plates of electropherogram data are, as FSA or HID files, are loaded up in batches. The computer is asked very quickly a batched question, actually a set of questions for each of the evidence items, for how many contributors might be there. And it's all done in one step because it's very automated. Just analyze this data and separate out the mixtures. The computer solves the problems and gives genotype answers, which are stored on the database, and then automatic database match with likelihood ratios compares these separated genotypes and produces match statistics. And those match statistics direct the analysis. Whether a pair of genotypes are found by the database within a case, with a strong match statistic, or they're found between cases. The starting point before the DNA analyst even begins to seriously engage the data is there already is an identification that's been automatically made. So it's worth commenting on the difference between genotype and allele databases. So here are some of the differences. In the left column, I've listed the attributes that we can take a look at, and the next two columns are for a genotype DNA database, and on the right, an allele DNA database. How do they handle mixtures? That's an interesting attribute. Genotype databases have a goal of separating the mixture into individual contributor genotypes, whereas allele databases use thresholds to keep the mixtures together, unseparated. How much can be uploaded? With a sensitive and specific genotype databases, 100%. All the mixtures can be uploaded, whereas in current practice with allele databases, maybe 10% of the mixtures can be uploaded. What kind of records are kept on the database? Well, genotypes are recorded as probability, whereas alleles are recorded in lists. And comparisons are made from genotype to genotype, whereas by having lists of peaks over threshold that are standing in for the data of the whole sample, the comparison on allele databases is being made between all the data from one EPG to all the data on another EPG as long as the peaks are over threshold. That's a data-to-data comparison. The comparison itself for genotypes computes a likelihood ratio in a genotype database, which is a measure of information between the evidence items and their contributors, whereas the allele comparison is an inclusion, an intersection, one of these sorts of allele database operations depending on the stringency level. The purpose of a genotype database is to preserve information. That's why it's created, whereas allele databases are not that interested in information, and so it can be discarded. The output of a genotype database are matches that are statistical with a strength, whereas the output in an allele database are investigative leads. Now, how many genetic loci do you need to get good results? With a genotype database that separates out mixtures, preserves information, uses likelihoods ratios, and so on, the 13 CODIS loci are fine, and the studies that we and others have done confirm that. However, if you use an allele database that loses a lot of information, because that's not its point, it's a generalization in a different direction from the 20th century, then you need more loci, and currently a lot of resources and money are being expended to expand the number of genetic loci in order to help support these older databases. What about false hits? If the method is sensitive and specific on a genotype database with likelihood ratios, then there are a few false hits. But there can be many false hits if the uploads aren't filtered on an allele database, and that can lead to a lot of cost, particularly to law enforcement and crime labs, as leads have to be chased down and hits have to be confirmed. In order to address that, what is uploaded is filtered by regulations. And so now we can look at the missed hits in practice. What are we not finding? On a genotype database, if everything's uploaded and it's an information system, you're not missing very much. But on an allele database, if you have to filter the uploads to just 10% in order to avoid false hits, then 90% of your evidence is not getting up to your database, and you're missing most of the potential hits. So there are drastic differences, regardless of the application, between information-based genotype systems and lead-based allele systems. So that concludes the fourth talk in the How TrueAllele Work series. Part 1 was on genotype modeling and the likelihood ratio back in October. We then moved on to degraded DNA and allele dropout. Last month we talked about kinship, paternity, and missing persons. Today the focus was on genotype database and DNA investigation. And these talks and webinars are available on this webpage at Cybergenetics for webinars. So with that, I will take any questions that people may have.